Hello and welcome back to the Biochemistry for Health Sciences channel. In today's video, we shall talk about DNA replication using E. coli as a model. In a previous video, we talked about the cell cycle, and we mentioned that for human cells, DNA replication or DNA synthesis occurs in this part of the cell cycle called the S phase. However, before we talk about replication in humans, let's look at replication in a simpler system, and that is the bacteria E. coli. Now, we know much more about E. coli replication rather than human replication. One of the reasons for this is because the E. coli bacteria has only one chromosome. It's a very simple system with 5 million base pairs. Compare that to a human cell that has 46 chromosomes, 3 billion base pairs, and on the top of that, there is very complex packaging of the chromosomes in a human. So it's very challenging to study human DNA replication compared to E. coli. But fortunately, a lot of concepts that we see in E. coli about DNA replication are quite similar to what we see in humans. So understanding E. coli replication not only will help us better understand replication in human cells, but it also has another important advantage, and that is in the field of medicine. If we can understand replication or metabolism in bacteria, that will help us develop and understand the mechanisms of antibiotics. And this is important because infections is a huge area in the field of medicine. Let's first go through an overview before we get into the details. So the E. coli has one DNA. It's a circular double-stranded DNA. Replication starts at a certain location in the DNA called the origin of replication, represented as ORIC. There is a protein called DNA A that will bind the ORIC and then melt or separate these two strands at ORIC, and that will initiate replication. Once the two strands at ORIC are separated, then more proteins will come and bind the origin of replication, and these are proteins such as helicase, as well as several other proteins, including the very important enzyme DNA polymerase that copies the two DNA strands. And all of these proteins form what we call a replisome. The helicase will start unwinding the rest of the strands in both directions, and as the strands are unwound, the replisome will travel and copy the two strands, one replisome going in one direction, the other replisome going in the other direction. When the strands are copied, so in this direction the two strands are copied, and similarly in this direction the replisome copies the two strands, the two replisomes will meet, and replication is terminated at a region of the DNA called the fork trap region. Each bacterium cell will receive its DNA. So one bacterium here will receive the DNA. Another bacterium that is formed will receive its DNA. If you look at this DNA, one of the strands is the parent strand. That's a parent strand, while the other strand is the newly synthesized strand. So each cell will have a parent strand and a newly synthesized strand. Similarly, the other cell will have a parent strand and a newly synthesized strand. So this is called semi-conservative replication. So let's look at the key steps of replication, focusing primarily on the concepts as well as the key proteins that are involved. The origin of replication, shown here in red, is a single 45 base pair sequence, and we call that the ORI-C. The protein DNA A, together with the help of another protein called prokaryotic histone-like protein, or HU protein, these proteins will recognize 9 base pair repeat sequences. These are called DNA A boxes that are present in ORI-C. And these proteins will then bind the ORI-C. Now, ORI-C is very rich in AT sequences. So remember, A hydrogen bonds with T with only 2 hydrogen bonds, compared to G, which uses 3 hydrogen bonds to bind with C. So clearly AT is a weaker interaction. So because the ORI-C is rich in AT, it's a weaker region, and that allows the DNA A protein to bind and melt the ORI-C area much easier because it's weaker, and this opens up the ORI-C segment. So you can see one strand separated from the other strand due to melting by the DNA A as well as the HU proteins, and this creates what we call a replication bubble. After making this bubble, there is now a lot of space available between the two strands. We see another protein, a DNA C protein called a helicase loader. This protein will now load a helicase or DNA B on each strand. So we see a helicase on this strand, we see a helicase on the other strand. After loading the helicase proteins on each strand, the loader now leaves the ORI-C region. Now each of the helicase will use ATP energy and will start unwinding the two strands in opposite directions. So this helicase will travel in this direction, unwinding these two strands, while this helicase will travel in this direction, unwinding these two strands. So this region here that resembles a fork is called a replication fork. Similarly, there is another replication fork. So there are 2 replication forks created around the ORI-C. Now remember that this strand is complementary to this strand. So if we separate the 2 strands and do nothing about it, they will come together, they will re-anneal, they will rejoin. So in order to prevent the 2 single strands from rejoining and also prevent the single-stranded DNA from attack by nucleases, single-stranded strands are more sensitive to nuclease attack. So we have these proteins called single-stranded binding proteins, or SSB. So these SSBs will start binding each strand. And by binding each of the separated strands, they will prevent the strands from coming together, and they will also protect the strands against nuclease attack. Now, as the helicase unwinds the DNA, what happens is that it causes this unwinding of these strands will cause twisting in the portion of DNA that's not separated. So you can see these strands that are not separated around here, they will be twisted, and that tension will increase so much that this area will start wrapping itself around itself to form what we call positive supercoils. So these positive supercoils that are ahead of the replication fork, they will hinder helicase progress, and that will stop DNA replication. So before we go further, let's discuss a little bit about supercoils. As mentioned earlier, unwinding two strands of DNA by helicase will form supercoils ahead of the replication fork, and these supercoils will prevent further DNA unwinding, and that will stop replication. We experience supercoiling when we try to unwind the strands of a double-stranded rope that is clamped at one end. In this example, if we try to separate the two strands of the rope, we will see the formation of supercoils, and that will prevent further separation of the strands. Now, supercoils can be positive, which is basically overwound strands, or supercoils can be negative, which is underwound strands. Positive supercoils will hinder replication, while negative supercoils will promote replication, because in this case the strands are underwound, here the strands are overwound. There is a topic in mathematics called topology. So topology is the study of shapes that can be deformed without changing their fundamental nature. So here we see a double-stranded DNA that is relaxed, and here we see the double-stranded DNA that is forming or has formed a supercoil structure. So supercoil DNA, relaxed DNA without supercoils. So in both cases, the DNA chemistry is the same, it's just that the shape has changed. Now, relaxed DNA is less compact. Supercoiled DNA is more compact. So in agarose gel electrophoresis, where migration is influenced not only by the size but also by the shape, we see that although the molecular weights, the size of relaxed DNA and supercoiled DNA are the same, the shapes are different. Relaxed DNA has a different shape. Supercoiled DNA is more compact. So supercoiled DNA will travel faster than relaxed DNA. So that's one way to observe supercoiled DNAs, by using electrophoresis. There are enzymes such as a type 2 topoisomerase. So topo comes from the word topology. Isomerase, remember, is an enzyme, family of enzymes. And there's a type 2 topoisomerase called DNA gyrase. And this DNA gyrase will release the torsional stress by introducing negative supercoils. So DNA gyrase will introduce negative supercoils, and these negative supercoils will neutralize the positive supercoils that are formed as helicase unwinds their two strands. This enzyme basically works by creating a temporary double-strand break in the DNA, then passing a segment through the break and then resealing the double-strand break. Now there is a pharmacological connection here. We have antibiotics such as nalidixic acid. This is primarily used for treating urinary tract infections. This drug inhibits DNA gyrase. Now, DNA gyrase is not found in humans. So by inhibiting DNA gyrase, it prevents DNA synthesis as well as cell division in these gram-negative bacteria such as E. coli, Klebsiella, as well as Enterobacter that are involved in causing urinary tract infections. Okay, so now we have unwinding of the strands, and the positive supercoils are neutralized by negative supercoils, and now this replisome can keep on traveling in either direction. So let's continue to see what happens. We'll just focus on one replication fork. Clearly the same thing will happen at the other replication fork. So from now on, we'll just focus on one replication fork. So at this replication fork, the helicase will now recruit other proteins, including DNA polymerase 3 that we'll call Pol3, this one here. Clamp loader complex, we'll call that CLC, which is this one here. Sliding clamps that we'll abbreviate as SC, that's SC here. So we have helicase, Pol3, CLC, and SC. All of these proteins, together with a few others, will form what is called the replisome. So here we have a replisome at one replication fork. As mentioned earlier, similarly there will be a replisome at the other replication fork. Now the actual copying of the strand to make a new strand is done by the enzyme Pol3, DNA polymerase 3. So if the parent strand had an A, the new strand will have a T. If the parent strand had a T, the new one will have an A, and so on. If there's a G here, there'll be a C. If there's a C on the parent strand, there'll be a G on the newly synthesized strand. Now DNA polymerase 3 or Pol3 has two very important restrictions. And this is important to understand. One is it requires a 3' hydroxyl group to start the synthesis. The second is that it can only synthesize DNA in one direction, 5' to 3'. If you have forgotten about these directions, you may want to review an earlier video on nucleic acids. Let's look at DNA polymerase a little bit more closer. So the DNA polymerase will copy the parent strand. This parent strand is also called the template. This is the template for DNA polymerase that it will copy. So let's say it wants to copy this A. The A will be copied as a T. So what happens is a DTTP nucleotide will enter the active site of DNA polymerase 3. And to copy the A into T, you can see that the enzyme DNA polymerase will require a free 3' hydroxyl group. And this hydroxyl group will attack this alpha phosphorus, the one nearest to the sugar. And when these two react, the hydroxyl reacts with the alpha phosphorus. What happens is you see this T, it gets attached to the new strand, newly synthesized strand. And these two phosphates will come out as pyrophosphates, which will then break down to give you phosphate. And all this releases a lot of energy that will make the reaction very spontaneous. So initially, this 3' hydroxyl group will come from an RNA. We call that the RNA primer. So initially, this DNA polymerase will start copying the template strand using an RNA primer. Once the RNA primer, a short RNA primer is made, then we'll see the 3' hydroxyl group will simply come from the copied DNA strand. So that's one requirement for DNA polymerase 3. It requires a free 3' hydroxyl to initiate the synthesis of new DNA. The other limitation of DNA polymerase 3, or the characteristic of DNA polymerase 3, is that it synthesizes DNA only in one direction, 5' to 3'. And you can see that from this reaction. You can see the T getting attached here. So you can see the synthesis direction is 5', And now the T will provide a free 3' OH group. Okay, so another nucleotide will be attached to the T. So let's say if this was a G on the template strand, then the next nucleotide to come in would be a C that will get attached to the T through the sugars. And that's why the reaction, the synthesis only goes in this direction. Starting from the 5', it will always go towards the 3' direction. Okay, 5' to 3' synthesis. Note that the new strand, this newly made strand, has a direction that is opposite, must have a direction that is opposite to the parent strand. So here the parent strand is running 3' to 5' from left to right. The new strand must run, in this case, 5' to 3' from left to right. They have to be anti-parallel. So it doesn't matter which DNA polymerase we are talking about. Here we are looking at replication in E. coli. Later we'll discuss replication in the human nucleus, nuclear DNA, and then later the mitochondrial DNA in a human. And all of the DNA polymerases have these requirements. They must have a 3' hydroxyl group to do the synthesis, and they can only synthesize in the 5' to 3' direction. Now, copying DNA by DNA polymerase is a very accurate process. The error rate is, you can see, 1 out of 10,000 to 100,000 base pairs. What is the reason for this? Why is DNA polymerase and in general copying DNA so accurate? One reason for that is the active site of this enzyme. It can differentiate the geometry of base pairs that are formed by correct base pairings, that means AT with GC, from incorrect base pairings. For example, A with C or G with T. If the correct base pair enters the active site, then the active site very, very rapidly changes its shape, its conformation, thereby very quickly catalyzing the reaction, very fast catalysis. However, if the incorrect base pair comes to the active site, then the enzyme shape changes 10,000 times more slowly. So its catalytic activity is very, very weak, resulting in negligible catalysis of DNA synthesis. So this once again shows this very important concept that we have mentioned many, many times in previous videos, and that is the importance of shape in protein functions, in biological functions. In addition to its shape, polymerase III also has proofreading capability. The enzyme has a 3', 5' exonuclease activity. So if a wrong nucleotide was incorporated into the new strand, this 3', 5' activity of this enzyme will replace the wrong nucleotide with a correct nucleotide. And this increases the accuracy over 100 to 1,000-fold. In addition to the enzyme properties, the other reasons why its accuracy is very high, one of them is a balanced concentration of the deoxynucleotides. So, for example, the concentration of the purine nucleotides, the concentration, say, of G is more or less similar to the concentration of C. The concentration of A purine nucleotide is similar to the concentration of T nucleotide. So by having the similar concentrations, the chances of them entering the active site would be equal. In addition, there are repair systems, DNA repair systems, such as the mismatch repair system, and these can also correct errors. In case there is an error, the mismatch repair system can correct the errors. And this system, you can see, can reduce the error rate to 1 in 10 billion base pairs. And all of this is very important. The accuracy is very important because this is our permanent genetic information. So when we copy this information into the new cell, we want the new cell to have the same information as the previous cell. Okay, so with this background on DNA polymerase, let's continue the synthesis. So what happens now is a clamp loader complex, CLC, shown here. This will load a ring or a sliding clamp that will abbreviate as SC around this strand that is running from 3' to 5' direction. You can see it's running in this direction, 3' to 5', with respect to how the replisome is moving. The replisome, you can see, is moving in this direction from left to right. Now, why do we need a sliding clamp? The sliding clamp will help the polymerase, the DNA polymerase 3, to stay onto the template strand. So it will help it keep on the template strand, and it will, as the polymerase moves and copies the template strand, the polymerase will not fall off from the template strand. So this SC prevents polymerase from falling off the template strand as the polymerase activity continues. So before the polymerase 3 can start copying the template strand, as we mentioned, we need a 3' hydroxyl group, and this comes from a short RNA primer. So we see a short RNA primer, and this RNA primer is made by the enzyme primase or DNAG. And this primer will provide the 3' hydroxyl group necessary for polymerase 3 to start copying the template strand. So as the helicase unwinds the two strands, the polymerase 3 can copy this strand here that's running from left to right, 3' to 5', and you can see the synthesis, as we mentioned, the synthesis of the new strand from left to right is running 5' to 3'. So that means the new strand is anti-parallel to the template strand. So this is all very good. So in this case, this strand here is called the leading strand. You can see the leading strand will be copied continuously. Okay, so the DNA polymerase will continuously copy the leading strand. It will travel along the leading strand and keep on copying the leading strand. So the leading strand is copied continuously by the polymerase 3. And this is possible because the newly formed strand is running 5' to 3' direction anti-parallel to the parent strand, which is exactly what we want. Now we have a problem with the other parent strand, with this strand here. You see the replisome is running in this direction. It can only go in one direction. So in this case from left to right. But and synthesis, if the new strand was also synthesized in this case, the new strand would be running 5' to 3' left to right. Okay, so the newly formed strand is running 5', 3', but the parent strand that is being copied is also running 5' to 3'. So these are parallel strands. That's not possible. We cannot have parallel strands. We must have anti-parallel strands. So this means the strategy to copy this strand here would be different than copying the leading strand. So let's see what happens. So once again, the primase will make a RNA primer to provide the 3' hydroxyl group. And we see that right here, the primer is being made by this RNA primase. And this primer will provide the 3' hydroxyl group. The clamp loader complex will now load a sliding clamp onto the other parent strand. So here we see an SC, a sliding clamp, being loaded onto this parent strand. And once the sliding clamp is loaded, the enzyme polymerase 3 will now attach to this template, and it will start synthesizing or copying this template, but it will copy, remember it only copies in 5' to 3', which means it will copy here 5' to 3' from right to left. From right to left. Remember, the machinery is running left to right. And these strands here on this other parent strand, this newly synthesized strand, is running right to left in the 5' to 3' direction. So this is what we want, 5' to 3' for the template strand, 5' to 3' for the newly synthesized strand that is complementary or anti-parallel to the template strand. So this is good. So since the replisome is moving in this direction and synthesis is going in the other direction for this strand here, we can only copy this strand in fragments. These fragments are called Okazaki fragments, and they're about more than 1,000 nucleotides long. So this parent strand is called the lagging strand. So the lagging strand is copied as fragments that we call Okazaki fragments. After an Okazaki fragment is made, the polymerase 3 enzyme as well as the sliding clamp will dissociate from the DNA. So as the replisome keeps on moving left to right, unwinding more and more parent strand, the newly synthesized strand, the previously synthesized strand, will re-anneal to the parent strand to give you this duplex here, and similarly to give you this duplex, this DNA bound to the newly synthesized DNA, hydrogen bonding to the parent strand. So as mentioned previously, the leading strand is the leading strand. It is copied continuously, while the lagging strand is copied as fragments called Okazaki fragments. So since the leading strand is copied continuously, but the lagging strand is copied discontinuously, all of this has to be coordinated. The replication has to be coordinated between the leading strand as well as the lagging strand. And one way this is done is by pulling the lagging strand, making it into a loop. So basically looping the lagging strand. So as the replication machinery moves along the DNA, it will extrude a lagging strand loop to bring the synthesis site closer to the synthesis machinery. And this will allow both strands to be copied simultaneously despite opposite synthesis direction. This whole structure here resembles a trombone slide, and therefore this is sometimes called the trombone model of replication. The lagging strand will keep on getting copied. Again, a primase will make a RNA primer in order to start a new Okazaki fragment. More of the lagging strand becomes looped, and that brings the primer closer to the synthesis machinery. The sliding clamp is loaded onto the lagging strand, and now the polymerase tree will be able to extend the primer and to copy the template strand into another Okazaki fragment. Now, another enzyme called DNA polymerase 1 or POL1 will use its exonuclease activity to degrade the RNA primer and replace the RNA primer with DNA, because this enzyme also has polymerase activity. So the RNA primer is degraded and replaced by DNA. The Okazaki fragments are then joined together by an enzyme called DNA ligase, which basically seals the nicks between the Okazaki fragment, joining them to form a single continuous strand. So this process is repeated as the replisome moves along the DNA, copying both strands. How is DNA replication terminated? Termination occurs in the replication fork trap region, which is opposite the origin of replication. So in this replication fork trap region, this part here, we see these proteins called terminator utilization substance, or TUS proteins. These proteins interact with TER sites on the DNA, and that leads to the formation of multiple unidirectional fork barriers. So you can see these arrows going in this direction and these going in this direction. So these unidirectional fork barriers will allow the replication fork to enter but not leave. Similarly, this fork can enter, but it cannot overcome this obstacle. And this ensures that the replication forks will meet at the termination site, even if one fork is delayed compared to the other. There may be other factors that may be involved in replication termination, because it is seen that the opposing forks will meet even without TUS-TER systems. So they will meet and stop replication even without the presence of TUS-TER. So there must be other factors besides this TUS-TER system that is used for termination of replication. After the synthesis is completed, the product circles are catenated or interlocked. You can see this circle is interlocked with this other circle, and these interlocked circles are separated by a type 2 topoisomerase called topoisomerase 4 or topo4. Like DNA gyrase, topo4 also cuts both strands, passes a segment of DNA through the break, and subsequently seals the break, and that leads to the production of two DNA circles. So each cell will have one template strand and one newly synthesized strand, and the other cell similarly will have one template strand and a newly synthesized strand, what we call semi-conservative replication. So this is basically how replication occurs in bacterium E. coli. In the next video, we shall discuss replication in humans. So until next time, thank you very much for watching and supporting this channel. Have a wonderful day, and we'll see you in the next video.